So by now you've already become familiar with the three different types of bodily fluids that we've discussed before. Intracellular fluid, which is all of the fluid that's contained within all the cells in your body. Interstitial fluid, which is all the fluid that is outside of the cells but not contained within the blood vessels. And then, of course, the blood plasma itself. So what keeps these three different bodily fluids compartmentalized from each other is physical barriers. And these physical barriers are called plasma membranes, and they exist and surround all of the cells in your body. So now we need to take a look at what actually makes up all of the plasma membranes. So plasma membranes are made up primarily of a type of molecule called a phospholipid. So phospholipids have a very unique chemistry, and as we're going to see here in a minute, that unique chemistry is going to play a determining factor in the shape and structure of the plasma membrane itself. So if you actually take a look at the phospholipid here, you can actually see that there's three major components. So first, in red here, is a molecule called glycerol. So the actual structure of glycerol is not real important, but what you're going to see here is the presence of these three oxygens. These three oxygens are more or less attachment points for different types of molecules. A triglyceride, for example, is a glycerol molecule that is attached to a total of 3 fatty acids. However, what you're going to see here for the phospholipid is that we are attached to 2 fatty acids. So this is essentially a diglyceride rather than a triglyceride. So for a phospholipid, the question then becomes, what takes the place of that third fatty acid, where that third fatty acid ordinarily would be? So instead of having a third fatty acid, you instead have a phosphate ion. So if you keep track of the three major components here, we have the glycerol, the 2 fatty acids, and then the phosphate head. This is where the unique chemistry of the phospholipid is going to come into play, because the fatty acid tails and the phosphate head are about as different chemically as they could possibly be. On the one hand, the phosphate head is very polar, and therefore we say it is hydrophilic, meaning that it likes to interact with water. On the other hand, the 2 fatty acid tails are extremely nonpolar, making them hydrophobic. On the other hand, they do not like to interact with water. So essentially, we've got these phospholipids, which you can see here in this stylized form, where we have the phosphate head and the fatty acid tails. Essentially, you've got this molecule that is kind of being pulled in 2 directions. The head really likes to interact with water, the tails really do not. So since all of our bodily fluids are essentially made up of water, the question then becomes, how do we construct the plasma membrane? How do we construct these physical barriers so that the phospholipids can actually do what they're intended to do, but at the same time, we don't want to force the fatty acid tails to interact with water, since they clearly do not like to do so. So this is where the unique chemistry is going to determine the structure of the plasma membrane. The way that we get around the fact that the fatty acid tails do not like to interact with water is that they're not going to have to. So instead of using a single layer of phospholipids, we are going to use a double layer, what is called a bilayer. Using this as our approach, we can set up the phosphate heads, keeping in mind the phosphate heads are hydrophilic, meaning they like to interact with water. You can see here the hydrophilic heads are pointing out towards where the water is. In this case, this might be the fluid inside the cell. Out here, this might be the fluid outside the cell. So these heads are facing out towards the water, which we've already established they very much like to do, whereas the tails are pointed in towards each other. And the interior of this bilayer is going to completely exclude water, meaning that everyone is happy. The heads get to interact with water, the tails don't ever have to see water. The tails are basically forming a no water club where they are the only members and they never ever have to see water. So everyone in this arrangement ends up being happy. However, we talked about the plasma membrane essentially acting as a physical barrier. However, the plasma membrane is usually said to be selectively permeable. Essentially what that means is that certain things will be able to pass this barrier while certain things will not. So finally, the last thing we can do here, now that we understand the actual chemistry of the phospholipids and the way they are arranged in order to form a plasma membrane, we can talk about the rules that we apply to determine what gets through and what does not. So first, if you start up at the top here, we have a class of molecules that we can say are very small and also nonpolar. These are going to be the molecules that have the easiest time passing through the plasma membrane. They are very small, so they should be able to squeeze in between two contiguous phospholipids. On the other hand, because they are nonpolar, they have no issue interacting with the fatty acid tails, which are also nonpolar. So these are going to be the molecules that have the easiest time getting through. In general, these molecules will be things like gases like oxygen and carbon dioxide, and then physiologically speaking, these are also going to consist of steroid hormones like testosterone, aldosterone, estrogen, and molecules like that. The next class of molecules we're going to look at here are large macromolecules like nucleic acids like DNA and RNA, and then also very large proteins. In this case, the polarity of these molecules is inconsequential. The problem here is their size. These are extremely large molecules. They are going to be far, far, far too bulky in order to squeeze in between phospholipids. So that is a deal breaker, meaning that proteins and nucleic acids will never be able to pass through the plasma membrane. So in their case, size is the exclusionary factor. Moving down the list here, small polar molecules, as opposed to the small nonpolar molecules we looked at previously, small polar molecules are going to have a much harder time getting through. The two examples we're looking at here are water and ammonia. So these, rather than getting straight through like the small nonpolar molecules, these are going to be rebuffed. So the problem here isn't their size. These are absolutely molecules that are small enough to pass through the membrane. The problem here is their polarity. Because these molecules are so polar, and because the phospholipid heads are also very polar, these are molecules that are going to be repelled by the heads before they ever have a chance to make it into the hydrophobic interior. So molecules like water and ammonia, for the most part, are not going to get through, although there is a very small percentage of these molecules that will get through. But the point is, these molecules will not get through very efficiently. And then finally, the last class we'll look at are electrically charged species like ions like sodium, potassium, and chloride. In these cases, these are as small as solutes can get. These are essentially the size of atoms. So definitely the exclusionary factor here is not their size. They are definitely small enough to make it through. The deal breaker here, and the thing that really kills them in this case, is their electrical charge. The electrical charge of these species, like sodium and potassium being positively charged, chloride being negatively charged, this is an absolute deal breaker for them, and they will not be able to get through because they will be completely repelled by the phosphate heads on those phospholipids. So if you compare this to our small polar solutes, these are polar, so they're going to have a hard time getting through, but a very small percentage of them will get through. Those are actually not electrically charged, they're just polar. These, on the other hand, their electrical charge is going to absolutely prevent them from getting through. So here, to sum up, we've talked about the chemistry of the phospholipid itself, how that determines the structure of the plasma membrane, and you can see that we end up with this bilayer, ensuring that both the phosphate head and the fatty acid tails are both happy in the sense of what they do and do not like to interact with. And then we've looked at these different classes of molecules and the rules that dictate what gets through and what does not.